The overall goal of this procedure is to facilitate the rapid and reproducible precellularization of bioinks for bioprinting applications while preserving cell viability.
This method can help streamline and improve the reproducibility when blending a cell suspension with a bioink to ensure the printing of viable cells. The main advantage of this technique is that it eliminates variability in mixing by blending cells with a bioink in an enclosed system with minimal handling and risk of sample loss.
Visual demonstration of this method is critical as there is a risk of improper mixing if the assembly is performed incorrectly.
To begin, obtain 2 syringes. One syringe is for the cell suspension, while the other syringe is for the bioink. The mixing ratio utilized in this study is 10:1. Therefore, use a 12 ml syringe and a 1 ml syringe as required by the 10:1 mixing unit. Also, obtain a sterile passive mixing unit that can be coupled with dual syringes via a Luer lock connection and a sterile female-female Luer lock connector. Retrieve a dispensing unit that can extrude a volume from 2 sterile syringes simultaneously at a controlled rate. Finally, obtain a sterile cartridge to directly mix the bioink and cell suspension into. In this protocol, a nanocellulose-alginate-based bioink is utilized, which is cross-linked through the addition of a calcium chloride solution post-printing. To prepare the cells, detach them using a 0.5% trypsin EDTA solution.